In this video, we will discuss about a further enhancement of the anti-tumor response of the iCAR-TTCL using interleukin 15. So this iCAR-TTCL is basically the CAR T cells that are formed from mature T cell lineage from the iPSCs, differentiated from iPSCs, that is induced pluripotent stem cells. So first they tested it with three prominent cytokines, that is interleukin 15, interleukin 21 and interleukin 7, out of which this interleukin 15 gives a high proliferative results for the iCAR-T cells, and those results are in supplemental figures. Therefore they decided to use interleukin 15 in their final anti-tumor efficiencies experiments or studies. So the map of this interleukin 15 plasmid that is used for transduction in the iCAR-T cells are shown in the panel A, where we can see it has the interleukin 15 membrane bound interleukin 15 and also this interleukin R alpha doping, and thereafter they transduce this interleukin 15 into the iCAR-T cells and then compare its results with the iCAR-T cells without any modifications. And what they are able to see is that in terms of the proliferations, there is an increase in the proliferations when they use the interleukin 15, mouse interleukin 15 or recombinant human interleukin 15, and same results they have. However, there is not much increase they have observed for the CAR T cells that are derived from PBMCs, that can be seen here, and the results in the iCAR-T cells are also marginal, they are not statistically significant. However, these results are good enough, they good enough to proceed for the in vivo studies as thought by researchers, because they thereafter they did the in vivo studies where they inject the iCAR-T cells in the presence and absence of MBIL, and the plan is shown in the panel C, where we can see that first they inject this tumors GH7, and 3 days after that they induced these CAR T cells, and they introduce CAR T cells at day 0 and day 7, and then check the retention times of the CAR T cells and its anti-tumor efficiencies effect. In terms of the retention times, we can see that iCAR-T cells along with the presence of MBIL shows high retentions compared to the case of iCAR-T cells without interleukin 15. These things they have quantified in the panel E, where we can see that even after that day 7 the increase is statistically significant, and the day 49 day 11 is increased further this difference further increased, and at day 19 they maintains that increased, so indicating that retention times increased within the CAR T cells. Retention time increased when the CAR T cells are present with interleukin 15, and they are able to sustain that increased for a longer period of time. And when they check it about the anti-tumor efficiencies effect, they are able to see the similar results, that is when the iCAR-T cells are used along with this mouse interleukin 15 antibodies or mouse interleukin 15 cytokines, they are able to see high tumor suppressions, and this increases statistically, and this increased suppression is statistically significant compared to the case when they see the results for iCAR-T cells without interleukin 15 antibodies. So this is how, and some of the results are also shown in the supplemental figures, where they did the comparisons also for this CAR T cells that are derived from PBMCs, that is from the patient's blood directly. So I hope you understood how they check or how they further enhance the anti-tumor efficiencies of iCAR-TTCL using interleukin 15, and why they come up to this interleukin 15, not other cytokines. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description, and there is also a brief gist of the results provided in order to understand these results in a more precise manner. So thank you for your time.